Good morning and good afternoon to everyone. I am Evelina Parisi from Eurofins Tecna sales department. Eurofins Tecna was founded 25 years ago and today is the competence centers for mycotoxins in Eurofins Technologies division. The two-day webinar will hold by our product manager, Giulia Rosa. Please, Giulia.
Thank you very much, Evelina, and thank you to all the participants for this connection this morning and this afternoon for the Asiatic attendees. I am very glad today to provide you this presentation on Aflatoxin M1 screening solutions from Eurofins Technologies. Before we start, I would like to let you know that you are all muted, but I do encourage to type your questions to my colleague Evelina in the chat box. At the end of the presentation, I will be very glad to answer your questions. I will make today a short presentation, a short introduction on the main sources of aflatoxin contamination in milk, having an overview over local jurisdictions, and I will present then iScreen AFLA M1 milk ELISA kit. That is the world first AOAC international approved screening method for the quantitative research of aflatoxin M1 in milk and milk powder. Being, well aware of the routine challenges of laboratories, I am very glad to present also an innovative automatic platform for ELISA implementation. That is, in my opinion, a very interesting option to standardize the performances, save time, and for the time being also to maintain some social distancing between people. And finally, I will conclude providing an overview over the available options from Eurofins Technologies for the analysis of aflatoxin M1 in different dairy matrices, covering also different purposes. So let us start with the metabolic production of aflatoxin M1. We all know, that molds grow mainly over vegetal commodities under wet and hot conditions. Nevertheless, drought is a source of stress for plants, and drought can lead to the reduction of their capability to defend themselves against fungi. So mycotoxins, and especially aflatoxins production, is boosted under dry and hot conditions. For instance, during rainless, hot summer seasons. Aflatoxins are typically on-field toxins that contaminate maize mainly, among many other vegetal commodities like fruits, nuts, and spices, other cereals, and so on. But let me mention that aflatoxin growth can occur also on stock silages at the end of the storage time, let's say usually close to the novel harvesting season after 1 year of storage in foxholes and silos. When cows are fed with aflatoxin B1 contaminated maize, they do metabolize the native substance into a more hydrophilic compound by inserting, you can see here in the bottom of the slide, an oxidryl residue in the molecule. And this more hydrophilic compound is named because aflatoxin M1 because of M like milk. And indeed, it is excreted in milk with a carryover ratio that depends on many factors, the health of the cow, the age, and so on, the concentration of contamination of feed. It was thought to be a ratio around 1%. Now it has been demonstrated that it ranges between 2 and 6%. Although the purpose of the metabolic path of cows is the detoxification of native toxin, you all know that Aflatoxin M1 is still classified as cancerogenic. And this is the reason why we have in place different jurisdictions and different regulation in the world. The world is actually divided into two main groups. On one hand, we have the European Union that established that the maximum aflatoxin concentration in milk at 0.05 micrograms per kilogram of milk. That means 50 nanogram per kilogram or 50 ppt. The limit is cut at 0.025 micrograms per kilogram of milk for infant formulae and dietary foods for infants with special purposes, special needs. The same levels were adopted by neighbor countries. These levels are very low, some of the lowest that I have ever seen for chemical contaminants in food. This is because milk is intended as a food for infants in general, and our exposure to it is pretty wide. Neighbor countries sometimes have modified the limit over the time to match with their contingent contamination and to protect the domestic production. It was the case of Serbia in 2014, where the dramatic outbreak of aflatoxins in cereals and aflatoxin M1 contamination in milk led to a temporary modification of the limit up to 0.2 micrograms per kilogram. So it was allowed to have a four times higher contamination. I have to mention that also some European countries during the outbreaks in 2012, for instance, demanded to the European Union to have temporary modifications of aflatoxin M1 limit in milk to prevent their production. They had no success. It was not allowed to have this temporary modification. Their request was based on the fact that for the rest of the world outside the European Union, the limit is as high as 0.5 micrograms per kilogram. The Food and Drug Administration limit is indeed 10 times higher than the one that is in place in Europe. And this level has been adopted by the US and Canada, Latin American countries, apart from Chile, China, and many of these other Asiatic countries, and most of the Arabic countries. So we have this split situation, split and different situation around the world. Let us enter in the very core of this presentation. Let me present now you iScreen AFLA M1 Milk ELISA kit. This kit is the world's first AOAC international performance-tested method for the quantitative research of aflatoxin M1 in milk and milk powder. The assay was actually developed around 20 years ago here in our laboratories in Italy. Sorry. We followed ISO 14675-2003 prescriptions. Sorry, let me take some water. The kit was designed to analyze raw milk directly after milking within 24 hours, but it can also be stabilized with preservatives and stored for delayed analysis. It is also possible to run the milk directly in the assay as it is with natural content of fat, or process the sample with a centrifugation step to remove the fat as prescribed by the ISO guideline. The assay is 25 minutes long, consists of 3 incubations. The first incubation is the binding reaction between the antibodies coupled on the solid phase and the M1 molecules in the standards or samples. The second incubation is the competition of the enzyme conjugate for the same binding regions of the antibodies. And finally, in the third incubation, the colorless substrate that is added into the wells is converted into a blue product. The color intensity after a short development time is inversely proportional to the aflatoxin concentration of standard N samples. By adding the stop solution, the reaction is blocked and the color changes into yellow. Results are obtained by means of a reader equipped with a filter at 450 nanometers. Data can finally be calculated by means of the spreadsheet that is available for free at our website. We include everything that is needed for the analysis inside the kit, including also 7 standards that cover the range 0.005 to 0.25 micrograms per liter of milk. Why do you include 7 standards? Because, as I was mentioning earlier, the kit was projected since the beginning to match with the ISO guideline. These tables compare the list of requirements from the ISO guideline in terms of everything: performances, sensitivity, selectivity of the assay, curve positioning, kit components, nature of the antibody, the conjugate, and so on, cross-reactivity to M2 with the iScreen AFLA M1 milk features. And you see that all the requirements are perfectly fulfilled. We're using polyclonal antibody, Aflatoxin M1 conjugate. We can offer the 96 wells plate format and the other 2 options in terms of size of the kit. The assay time is pretty lower than the limit. Internal specification of, let's say, the top and the mean value of the curve are perfectly in line with the requirements. Cross-reactivity to M2 is lower than 20%. We are including 7 standards that need to be run in duplicate. The calibration range is wider than what is requested by the ISO. Samples also are to be run in duplicate, and the sample dilution is permitted to match also with the FDA limit, and also to calculate the concentration for extraordinary highly contaminated materials. We have different options for the curve preparation. The repeatability of the standards is lower than the ISO prescription, and during the AOAC validation turned to be even lower. Limit of detection and limit of quantification were pretty much lower than what is requested as a limit in the ISO. The ISO requests for sample preparation for milk to run the centrifugation and the defatting, and this is one of the options for raw milk. And obviously, for milk powder, the reconstitution with water is requested, and recovery is always higher than 80%. This is the AOAC certificate that was achieved earlier this summer by my colleagues from the R&D. This certificate can be downloaded directly from the AOAC website. It includes a few pages with the product presentation, the validation discussion and results. And today now, I'm going to provide an overview of the validation protocol we followed, and I'm showing the results that we obtained here in Eurofins Tecna laboratories, and those also that were confirmed by a third-party entity that was an independent laboratory in the US named QLab that we didn't know before this trial and that used the kits for the very first time during this study. So, as a first step, Eurofins Tecna researchers ran the calibration study. We were asked to demonstrate the repeatability of curves. We ran 6 curves. They were obtained in different days by different operators, using also 2 readers. The table here shows the inhibition values of each standard obtained for each of the 6 curves. We have the mean results, and what is extraordinary in my vision is the coefficient of variation and the standard deviation and RSD coefficient of variation. Data show an excellent intermediate reproducibility, and indeed, you see in the image in the graph, here we have 6 overlaid curves. The shape of the curve is also very good, with a long linear range that is the key for having accurate and precise results. There are no flat portions at the beginning or at the end of the curve that are typical attempts to extend sometimes a measuring range that is naturally shorter. And this is just a way to confuse end users, pretending that the kit is able to detect something that is not capable to do, or maybe it can detect, but it cannot quantify with reliability. The second task was pretty complicated. We were assigned by AOAC for the AOAC verification was the investigation of the method selectivity. It's complicated in the sense that it was a complex and detailed study. The question is the kit fully specific to the detection of Aflatoxin M1 only? What if other compounds are in the sample? Can we wait for any interference from them? Maybe some overestimation that are so typical for ELISA methods? So we started with a negative milk that was firstly verified with LC-MS analysis, and it was controlled with ELISA and turned to be negative. We spiked the material with 100 nanograms per liter of Aflatoxin M1 and 100 nanograms per liter of M2. 100 nanograms per liter means 0.1 micrograms, so double times the European limit, a pretty high concentration. While the ELISA results for the M1 toxin was accurate, having 97% recovery, we weren't able to detect the M2, that is the closest molecule to Aflatoxin M1. And even if it is a relevant concentration, the assay was not touched by this event. The blank materials was then used for spiking experiments where we fortified 10,000 nanogram per liter of native aflatoxins, fumonisin, vomitoxin, ochratoxin, zearalenone, sterigmatocystin. The aim was to stress the method with very high concentration to confirm its specificity. And the assay indeed returned unmodified results. So the blank material was still detected as blank for Aflatoxin M1. We had a minor detection of B1 and G1 that indeed are pretty similar to Aflatoxin M1 in terms of chemical structure, but the interference was less than 0.2%, so nothing at this very high level. The last experiment was the verification of M1 dosage in co-presence with native toxins. So we kept this 100 nanograms per liter of M1 spiked sample, and we mixed with 10,000 nanograms per liter of native toxins. We created, for this exercise, a totally unrealistic environment. possibly due to a hook effect because it's too much. A negative interference was found when aflatoxin M1 was mixed with the native aflatoxins only, no interference from the rest of the molecules. We reduced the concentration of B and G aflatoxins to similar concentration to M1. It is still an unrealistic situation because it would mean that the metabolism of cow is not existing and the interference disappeared. So the conclusion was that there is no relevant interference to the dosage by any compound that realistically could be co-present in milk. And we never, ever found any higher recovery overestimation due to the presence of other compounds than aflatoxin M1. Entering now in the very heart of the study, the performances of the assay were assessed for milk, obviously. Samples were spiked at 5 different concentrations along the whole measuring range and were analyzed following 4 different procedures, as indeed, on the kit insert we mentioned that the milk can be analyzed as fresh within 24 hours after milking, or stabilized. And for both situations, it is possible to run it as whole with natural content of fat or defatted by centrifugation. So we have 4 conditions, 2 readers, 2 technicians, 5 replicates of each material that were used as blinded coded. The tables you see here summarize the average results that we obtained for the 4 protocols. You see, all the time, we were able to have very accurate results, always having compact data series with a limited standard deviation. We are always guaranteeing a higher recovery than 80%. You remember it was the limit prescribed in the ISO guideline. And also, we can see that there are no important differences among the 4 protocols. So it doesn't matter whether you ran the analysis on fresh milk, on stabilized milk, on defatted milk, or on whole milk. These were the analyses obtained by our hands. Let's say we are the developers, the producers. We know everything about this kit. But what about an independent user? The metric study was confirmed outside our laboratory in QLab, and local American fresh milk samples were used to repeat the experiments following just 2 of the products of the protocols available. And you see that despite the presence of an ocean between us and them, very similar recoveries and excellent repeatability were found. To complete the metric study for milk, the limit of detection and limit of quantification were assessed by using, obviously, a blank material. Once we assessed the expected limit of quantification by calculating the mean response of blank materials plus 10 standard deviation, we wanted to confirm it by spiking the blank materials at a close concentration. This is the table with results we got for 10 replicates of blank materials run with the 4 conditions. The calculation here, you can see the mean value that we obtained following the 4 protocols, the standard deviation that was very compact. We calculated the limit of detection that is pretty similar among the 4 conditions and ranged between 3 and 3.7 nanograms per liter. And we were also calculated the expected limit of quantification. So we went for the confirmation by spiking at 5 ppt. Again, 4 different protocols. We spiked the blank materials and ran 10 replicates each. We found this situation, all the determinations were detected by the assay as positive with consistent results. So we were able to successfully confirm that the limit of quantification can be set at 5 nanograms per liter. Results were confirmed by the QLab in the US, always by means of local fresh milk, that in their hands gave also even lower matrix effect. So in their hands, the limit of detection turned to be even lower. We think it's safer to consider 3.5 nanograms per liter as a limit of detection and 5 as a limit of quantification for all the protocols. The matrix study was then run for the powdered milk. The samples were reconstituted with water and treated like they were liquid milk. So all the results here are reported for liquid milk. The limit of detection and limit of quantification were assessed similarly to fresh milk by confirming the limit of quantification with a fortification experiment. We obtained absolutely very similar results comparing, let's say, these metrics to the results we obtained for fresh liquid milk. So it seems that the process of producing milk powder does not affect the metrics in any manner. This is the mean value that we obtained for blank materials. So we calculated the limit of detection, turned to be slightly lower than 2 nanograms per liter. Limit of quantification at around 5. We spiked at 5, and we're able to obtain consistent, accurate results for the spiked materials. For milk powder, we had an advantage. We could assess the accuracy and precision of the assay by using reference and control materials that are available from different suppliers. Sorry. Here. And we were able also to cover with these materials the whole measuring range of the assay. Again, we are expressing the concentration in terms of reconstituted milk. So you see which were the expected values that were assayed by reference methods, so instrumental analysis. Then you see the mean results that we obtain, the standard deviation, and the average recovery. We obtained very good results here in Eurofins Tecna, and we were also able to compare with those obtained by the QLab. We shared the two of the materials, the blank and the lowest contaminated. Plus, they wanted to add one from Test Veritas company that is close to the European regulatory limit. And you see that they were always able to obtain successfully good results, very good recovery, and compact data dispersion. To finalize the AOAC verification, it was necessary to demonstrate the lot-to-lot consistency and the stability of the kit. Questions are the kit batches equal one to each other? Otherwise, it could be possible for us to have this verification just on one perfect batch. And also, are the performances of the kit equal during the time when it's freshly released by the production, at the middle of its shelf life, and close to expiration? To reach the goal, we used 3 different batches chosen at different ages. So freshly made half-life, and close to expiration. We ran the curves that always turn to be compliant and overlaid. Besides, a blank milk was used as negative and fortified together with a reference material, a powder milk, of course, reconstituted, with an assay value close to the European limit. It is clear from the linear regression reported in the graph that the assay keeps its performances despite the batch number and the age. The statistic comparison also of data collected for samples led to the demonstration that all data sets were comparable one to each other. So with this experiment, we successfully demonstrated the stability of the kit and lot-to-lot consistency. The very last aspect is the assay robustness. How is the response of the assay when the protocol is changed or the environment is different? We know that the incubation time could sometimes vary, especially when the analyst is, I don't know, overloaded with tasks. It's normal, for instance, to delay the preparation of the washing buffer, for instance, because you don't really realize that you have to have it ready. The temperature in the laboratory could also vary because, of the season or when the heating system of air conditioning do not work properly and so on. So as a result, we agreed with the UAC to apply factorial design to point out which of the differences introduced in the protocol, that is here in bold, you see the regular conditions that you should follow. And we investigated with this factorial design which of these modifications could lead to any performances deviation. So once more, we ran the calibration and used a sample also, a powdered milk sample. We spiked at a certain concentration, and the sample was run in 3 replicates, not to have one data only. As a result, the assay showed excellent results also when the incubation time was modified. Well, let me tell you, all the data were compliant. When the incubation time was modified, I have to say nothing really happened. Results were excellent, in line with the validation. Very modest influence from the temperature was recorded. It is, I have to say, really normal, considering the importance of the temperature of the kinetics of binding reaction. So this is normal. Still, the sessions were always compliant to specification. So this was the last experiment run within the AOAC approval. We were really satisfied. It is not a case that this kit would showed really excellent results on raw milk, despite the preparation, on milk powder, and we were glad to see the robustness of the assay, the consistency, and the stability. It is not the case, in my vision, that robustness is included in the verification protocol, and it is so important to have it. And I have to say that despite the excellent robustness of ice cream AFLA M1 milk, environment conditions could really affect the results of any immunoassay. At the same time, busy times, overloading days could also boost human mistakes other than those that we analyzed here. So Let me also reflect now on the urgent need to guarantee now on the workplace a proper distance between people. I am aware that this is fairly not easy to obtain and could lead also to further consequence on those running experiments if they are really overloaded with experiments. Why am I mentioning these points? Because we wanted to answer a question, is there anything we can do to support routine analysis of laboratories and industries, to standardize the assay execution and consequently the performances? Is there any way to free the analyst? Despite the quality of this very kit, is there anything that we can offer to support the routine of laboratories, of industries, and so on? So thanks to the Eurofins Technologies network of companies we now belong to, we're very glad to offer the analysts with different ELISA robots that are different because of their customizable throughput. They're characterized by excellent pipetting accuracy. They have integrated functions to carry the whole ELISA implementation without any external intervention. There is no need to take the kit, wash outside, or read in another reader. ELISA automated solution obviously are not an original invention, but compared to certain huge machines that were conceived at the beginning for clinical diagnostics and huge throughputs, I do believe that this very option that I'm here representing in this slide will meet the realistic needs of food laboratories. The Bolt is the most compact, easy to use and flexible ELISA robot. It hosts one plate, but multiple protocols could be run in parallel in the same frame if kits do share the same type of plate. It can be therefore adopted also for low and medium analytical volumes. In the past, let me mention that it was not reasonable. This is not a machine that has a meaning only for huge consumes. Which are the remaining, the other advantages? Being a closed system, it ensures a protected environment and guarantees, these guarantees obviously, excellent assay reproducibility over the time. It doesn't matter who is the person that loads the standards and samples in the racks. Doesn't matter if it's winter or summer. Everything is perfectly standardized. The Bolt belongs to Eurofins Technologies, which is the key point here, that it hosts many validated methods for different contaminants. Not just Aflatoxin M1, there are methods for native toxins in cereals, phycotoxins in water, pesticides like the glyphosate, residues of antibiotics and allergens. Also, there is the possibility to run the gluten ELISA test kit that is also AOAC approved, and it is based on the R5 Codex approved antibody. It is important to mention that the methods are already been validated because sometimes I know that these machines are attractive to laboratories, but then everything must be handled by the analyst. And before earning the advantage of having the automation in the lab, it is really a huge way having the training from the producer of the robot, then trying to have a program that runs the kit, and then you need to adjust the drift effect that usually happens with robots. You have to manage cross-contamination events and to adjust the performances in general. Everything is already done, so it's a reliable solution that immediately brings an advantage to the routine. Going back to Aflatoxin M1 in milk, the advantage of using the Bolt for M1 analysis in milk is dramatic. think that it is possible to analyze the milk samples without running any sample preparation. So it means that the person, the analyst, is really free. Once the materials are dropped into tubes and inserted in the robot racks, the analysis could be hands-run potentially by anyone with almost no training. And considering our current situation, this instrument looks to me as a real improvement to routine analysis with plenty of advantages. So it's robust, compact, helps in performance standardization. It comes with already validation methods that just need to be transferred from our laboratory to yours, and supports the distance in laboratory and free the people to do something else with, in the end, a very reasonable investment. We are reaching the end of the presentation. I just want to mention which are the options you have from Eurofins Technologies for the analysis of Aflatoxin M1 and different dairy products, and what you can choose to adapt to your real analytical throughputs. So this is the guy we widely discussed today about ice cream AFLA M1 milk. It's available in 3 formats: 48, 96, 192 determination. It means half plate, 1 plate, and 2 plates kit. It's interesting because it is possible to arrange a contract with us where you can receive a smaller format in low season and before, after harvesting. So when usually analysis increase, you can ask for the big format that helps you in saving money. Also, you can ask for the milk diluent if you want to shift the measuring range to adapt to be compliant to the FDA requirement. And also if you're in the need of diluting highly concentrated materials, you can have it. It's very cost efficient. If you want to run some spot application to cheese, then you can ask for the extraction buffers. Again, they are very cheap products. We can also release you the application notes. They are not AOAC approved, but you can run successfully on the kit. Also, there are 2 supplementary standards that have been conceived and released to boost the accuracy and precision around the European limit. So if you want to be very, very sure and confident about your quantification around the European legislation, you can add these 2 standards. And of course, we provide you also with the dedicated spreadsheet. The kit is ISO compliant, is AOAC approved for milk and milk powder, and the bolt application will be finalized this very month, so you will soon receive the information after our application launch. Should you have more cheese materials to analyze, then I would recommend to have the all-in-one kit. The ice cream AFLA M1 is the same kit, same reagent, same antibody, conjugate, assay time, everything is the same than the milk version. It does not have the USC approval just for, I mean, it was a choice since the beginning, but for milk, it works exactly the same as the milk version. But it includes, in the same box, the extraction buffer and the instruction to run the pasteurized milk, cheese and firm cheese with dedicated procedures, yogurt. We are releasing now the mozzarella cheese, and soon we will have other applications. And as for the milk version, we are launching this very month the Bolt application. What if the analytical volume of your laboratory, of your industry is low? the expense of running 7 standards in duplicate, you know, could be really dramatic, really important when you have in your routing a few samples to run per sessions, and milk is not easy to store for a long time. So you can see from the graph here, if you have 3 samples to be run per session, with a regular ice screen, you have to spend 14 wells for the calibration and 6 wells for your samples. It means 20 wells, pretty a quarter, around a quarter of the kit, just for 3 samples. The B0 is a master curve calibrated assay. It is specifically designed for low analytical volumes, and it includes just one standard, the 01, the uncontaminated, let's say the blank. You have it to run. You have to run it in one replicate only. Plus, you have to use one well for each of the materials of the samples you want to run. So, if you compare these volumes, here you are running 20 wells, and here just 4. The saving is dramatic. Still, the measuring the assay is quantitative. This is the measuring range, goes from 0.01 up to 0.2 micrograms per kilogram of aflatoxin M1 in milk. It is slightly less sensitive, but sufficiently sensitive for the European market. And there is another advantage, that is the assay time, because this kit takes just 30 minutes. I was mentioning that the measure is still quantitative. How is that possible? Thanks to the robustness of the assay and the condition of the reaction, we were capable to remove the standards from the kit as, real standards, real bottles that you have to run. And we provide the master curve as virtual in the kit certificate. So you can use that virtual curve that is assessed by us at the moment of the production for the calculation of your data. And I think it's really an advantage when you have to handle a limited number of samples per time. The Bolt application is already available. So you can have also the B0 AFLA M1 run in the Bolt. I want to take a moment to present my team. These are the ladies that we have in our laboratory, our R&D competence center in Italy. The company has, as Evelina was mentioning, more than 25 years of experience. Our science orientation is strong thanks to not to us just, but to the partnerships that we successfully had over the time with laboratories, many public institutes, and also dairy factories that cooperated with us and supported us and permitted us to learn what is the real need of a routine laboratory. And this led to our reference in the dairy sector. But also, it's not just a matter of Tecna. Now we are a part of a multinational network, so we are achieving a very strong support from Eurofins Laboratories and Eurofins Technologies network of partners. Just thinking to the Bolt that is available from one of our partner inside Eurofins Technologies. This is a consistent advantage in my vision to have us as a partner for your routing analysis. So thank you very much for your attention. And I don't know if there are any answers before, any questions, sorry, I hope so. Before we start, I would like to leave you my personal email address for any curiosity you may have in the future, and also the shared mailbox that is covered by my colleagues Valeria and Julia that are glad to provide you any technical assistant will respond to your questions before, when using the kit, and also after for any doubt you may have. Also, I invite you to visit our YouTube channel because there is video dedicated to the M1 kit, so you can learn how it works, which are the steps, and that's it. Evelyn, I don't know if you have collected any questions.
Yeah, thank you, Julia, for the clear and very detailed presentation of this morning. And yes, we had some questions from the audience. So the first one, ready?
Yes, absolutely.
Which is the sample treatment for pasteurized milk?
Pasteurized milk is can be analyzed directly in the assay without any manipulation. So you just bring the milk and run in the assay, or eventually, if you do prefer, you can centrifuge. It's like the raw milk, let's say.
Okay, thanks. So always remaining on the sample preparation, which is the difference between cheese and firm cheese?
Cheese are extracted in solvent using dichloromethane. Then you have to run, of course, the evaporation of the solvent. You need to reconstitute with buffer, and then you have to defat with hexane. For firm cheese, that is you know, like in Italy we have plenty of them, grana padano, parmigiano, and so on, seasoned cheese. We added a digestive step with pepsin. So these are the differences. Everything is included in the kit insert of ice cream AFLA M1.
Okay, thank you. Ice cream AFLA M1 milk is the only AOAC approved for ELISA method at the moment?
Yes, it's the only one that is approved AOAC. Yeah, for milk, raw milk, and milk powder.
The software for the interpretation of the results is available?
Yes, you can download directly from our website at the bottom of the product page. It's an Excel spreadsheet. It's for free. You type the absorbances you collect from your reader, from the calibration and for the samples, and you easily calculate the concentration. The spreadsheet for the milk version includes just the application to milk and milk powder. On the one for cheese, the M1, let's say, comprehensive version includes also the columns for calculating the results for cheese, yogurts that have different dilution factors. Let me tell you, Evelina, that it is possible to use any software included in each of the ELISA readers. Usually it's very easy to program, but of course also this copy and paste inside our spreadsheet is recommended and is dramatically easy.
Thank you, Julia. Why didn't you compare the assay results on milk with the LCMS?
Sorry, I didn't understand.
Why didn't you compare the assay results on milk with LCMS?
Because, well, we agreed, we had a long discussion with AOAC about how to run the validation on milk. Our question mark was how to handle this matrix that is not stable, like, I don't know, cereals or other vegetal dried commodities that can be stored and reanalyzed and so on. So we decided to have the comparison of the performances to LCMS when using the milk powder, the reference materials. For the rest, we agreed just to run the spiking the spiking experiments on raw milk for AOAC. Let me comment that, of course, this what I presented today belongs to the AOAC approval path and is the discussion, let's say, the presentation of the data that are attached to the AOAC approval and certificate. But in the past, also by the precious collaboration with plenty of laboratories and dairy industries, we made a lot of these comparisons. Verifying the correlation of our results to LCMS or HPLC analysis for Aflatoxin M1. It was actually the core of our validation that we had in the past. We still have those data. Some of them have been published also in some conferences and posters. So we did it in the past. For this very study, we decided not to do so.
Perfect, thanks. Since we are running out of time, I will read you one last question, and then I will strongly encourage for further questions to write to the address that you can see on these slides. For Thailand, most customers like to use rapid tests. So what should be the benefit of ice cream over those rapid tests, and which may also have AOAC?
There is a huge difference between the ice cream and the rapid test in many sense. I have to say that if rapid tests are used for I don't know, good acceptance or on-field analysis, they are really, they cannot be how can I say, replaced by the ice cream. I see maybe the B0 ELISA more suitable because it allows to get the results in 30 minutes. But of course, the ice cream do really have excellent performances. So the difference are in terms of accuracy, precision, sensitivity usually. So I think they fit for different purposes. In some of the practice that I know about, the two techniques can live together and you can be used for different purposes. So first, maybe I use rapid tests, I mean lateral flow devices, immunochromatographic assays for fast semi-quantitative analysis, and then I use the ELISA for let's say further analysis. Also, let me comment that the throughput of the analysis is different. So it's pretty hard to imagine to me that you can handle efficiently dozens of samples in parallel with strip tests. So also what I could imagine is that you can use the strip test for the analysis of, I don't know, mass commingled milk when you receive, I don't know, a tank or a track. But when you really want to learn something more about the farms, the different producers or the different samples that were mixed together in that tank of commingled materials, you have to use the ELISA. If you think that in a little more than 1 hour, you get the results up to 40, I don't know, samples for one plate, that is less than 2 minutes per sample. Let's say if you think to run one after each other. So I think that the two techniques cannot be interchanged, but there are some points for a fruitful discussion in my vision.
Yes, of course. Thank you. So Julia, to answer this session of questions, since the time is almost over, we strongly encourage you to write to Julia and to our colleagues of support and technical assistance. And please also visit our website and our YouTube channel, in which you can find a small video regarding the products. Thank you to all for your time and for your attention today. And we wish you all a very nice day. Thank you. Bye.
Thank you. Thank you very much for your attention. Thank you, Evelina, for your support. Once more, feel free to contact us for any requests or curiosity you may have. Bye-bye.
Goodbye.